immortal human cell line known as HeLa cells. Henrietta Lacks. Henrietta Lacks, 1920 to 1951, was an African-American mother of five whose cancer cells, taken without her knowledge or consent, became one of the most important tools in medical history. Known worldwide as HeLa cells, they were the first immortal human cell line grown in a laboratory. While her cells laid the foundation for modern medicine, her story sits at the center of the ongoing debate over bioethics, race, and patient rights. Who was Henrietta Lacks? Henrietta Lacks was born in Roanoke, Virginia, and later lived in Turner Station, Maryland. In early 1951, she went to Johns Hopkins Hospital, one of the few hospitals in the region treating Black patients at the time, for severe abdominal pain. She was diagnosed with aggressive cervical cancer. During her radium treatments, tissue samples were taken from her tumor without her knowledge or informed consent, a standard medical practice at the time. She passed away later that year at age 31, but her story was far from over. What made HeLa cells special? Before HeLa cells, A. Scientists struggled to keep human cells alive in lab cultures for more than a few days. They would inevitably age and die. Henrietta's cells had a rare mutation that allowed them to divide indefinitely as long as they were supplied with nutrients. This immortality enabled scientists to conduct standardized, repeatable experiments on human tissue for the first time. Key impact: HeLa cells have been cultured continuously since 1951, yielding thousands of tons of cells and contributing to over 110,000 scientific publications. A. Because HeLa cells were the first human cells capable of dividing indefinitely outside the body, they became a biological standardized workbench. 1. Polio Vaccine Development. 1950s. The breakthrough. Jonas Salk developed a promising polio vaccine, but testing it on millions of monkeys was prohibitively expensive and slow. HeLa's role: HeLa cells were highly susceptible to polio and replicated quickly. The National Foundation for Infantile Paralysis established a large HeLa cell manufacturing facility at Tuskegee University, using the cells to conduct large-scale safety and efficacy testing of the vaccine, which led to global eradication efforts. 2. Chromosomes and the Human Genome, 1953. The breakthrough. For decades, scientists believed humans had 48 chromosomes. HeLa's role. Geneticist Joe Hin Tjio used a special hypotonic salt solution on HeLa cells, causing their nuclei to swell and their chromosomes to separate cleanly. This technique enabled researchers to count 46 human chromosomes. A. Laying the groundwork for diagnosing conditions such as Down syndrome, trisomy 21.3, HPV and cervical cancer prevention, 1980s. The breakthrough. German virologist Harald Zerhausen hypothesized that cervical cancer was caused by the human papillomavirus, HPV. HeLa's role: by examining HeLa cells, Zerhausen discovered that they contained HPV-18 DNA embedded directly in their genome. This proved a direct link between viral infection and cervical cancer, earning him a Nobel Prize and leading directly to the creation of the life-saving HPV vaccine, Gardasil. 4. Advanced gene therapies and biotherapeutics. The breakthrough. Modern gene therapy relies on adeno-associated virus, AAV vectors, harmless virus shells engineered to deliver corrective genes into human cells. HeLa's role. HeLa cells are often used as biological factories to produce these viral vectors at scale. They have played a pivotal role in testing and manufacturing cutting-edge treatments such as Zolgensma, a spinal muscular atrophy gene therapy, and Kymria, a CAR T-cell therapy for cancer. 5. Virology, herpes and COVID-19, HIV to AIDS, 1980s. Researchers used HeLa cells to understand how HIV infects immune cells, discovering that introducing the CD4 receptor enabled the virus to bind to cell surfaces. COVID-19, 2020s. HeLa cells engineered to express the ACE2 receptor were used to study how SARS-CoV-2 invades tissues and to rapidly screen antiviral drug candidates and vaccine formulations during the pandemic. 6. Space medicine and radiobiology. The breakthrough. Before sending humans into space, scientists needed to understand how cosmic radiation and zero gravity would affect human tissue. HeLa's role. In the late 1950s and 1960s, HeLa cells were launched into orbit aboard Soviet satellites and NASA's Discoverer XVII mission. Scientists found that HeLa cells multiplied even faster in microgravity, helping establish radiation safety limits for future astronauts. Henrietta Lacks' story fundamentally changed the moral and regulatory landscape of modern biological research, exposing massive gaps in patient rights, informed consent, and health equity. 1. Informed consent and tissue ownership. When doctors took tissue from Henrietta in 1951, taking biological samples during procedures without consent was standard practice. Today, that is illegal. In closing, Henrietta Lacks never knew that her cells would change the world. But her unrecognized sacrifice permanently elevated ethical standards in medicine and saved millions of lives through scientific innovation. The common rule. Henrietta's case helped shape federal ethical regulations for human subject research, revised extensively in 1981 and 2018. It mandates that researchers obtain explicit informed consent before collecting biospecimens for research. Property rights over cells cases like Moore v. Regents of the University of California, 1990, established that individuals do not maintain biological ownership of discarded tissues. But Henrietta's legacy pushed the conversation toward requiring upfront commercial disclosure if tissue might be monetized. 2. The 2013 NIE Genome Control Agreement in German scientists sequenced and publicly published the complete genome of the HeLa cell line. The Lacks family was not informed, raising severe privacy concerns because Henrietta's genetic blueprint reveals health information about her living descendants. The outcome. The National Institutes of Health, NICH, met with the Lacks family and established a historic agreement, the HeLa Genome Data Access Working Group. Researchers who want to use HeLa sequence data must apply for access, and 2 members of the Lacks family sit on the review board that grants or denies permission. 3. Modern corporate reckoning and unjust enrichment lawsuits for decades. Pharmaceutical companies made billions off products developed using HeLa cells while her family lived in poverty without health insurance. That paradigm has shifted dramatically in recent years through high-profile legal action led by civil rights attorneys. Thermo Fisher Scientific, 2023. The Lacks estate reached a historic confidential settlement against Thermo Fisher for unjust enrichment from mass-producing HeLa cells long after knowing their non-consensual origins. Novartis and Viatris, 2026. The state reached confidential settlements with pharmaceutical giants Novartis and Viatris regarding their commercial usage of HeLa-derived therapies, including CAR T and herpes treatment. Ongoing litigation, lawsuits remain active against other biotechnology firms such as Ultragenyx over using HeLa cells to manufacture viral vectors for gene therapies. 4. Legislation and institutional restitution, Henrietta Lacks Enhancing Cancer Research Act. 2021, signed into United States federal law. This bill directs government agencies to review and reduce barriers to clinical trial participation for underrepresented racial and ethnic minority groups. The Henrietta Lacks Foundation, established in 2010, it provides financial grants for healthcare and education to individuals and families who were used in medical research without consent. In closing, Henrietta Lacks never knew that her cells would change the world, but her unrecognized sacrifice permanently elevated ethical standards in medicine and saved millions of lives through scientific innovation.